Collenchyma tissues. Collenchyma is a living tissue composed of more or less elongated cells with thick primary non-lignified walls. It functions as a supporting tissue in young growing organs and in mature organs of herbaceous plants. It is absent in woody stems, dolichots, which have undergone secondary growth. It is also absent in leaves and stems of many monocots, where sclerenchyma is appeared as an early stage of development. It is plastic in nature and stretches irreversibly with the growth of the organ in which it is present. The plasticity of collenchyma changed with the age. The mature collenchyma is less plastic, harder, and more brittle than breaks earlier young collenchyma. There is a physiological and morphological relationship between collenchyma and parenchyma. In place where two tissues are present side by side, transitional forms are observed between a typical parenchyma and a typical collenchyma. Plant exposed to wind, the walls of the collenchyma cells become thick-walled. It observed that mechanical shaking of the plants result in an increase in the amount of wall thickening of collenchyma cells. The collenchyma cells may be lignified and the walls become thick and resulting in the formation of the collenchyma. The photograph shows here the different types of the collenchyma are produced in different types of the plant organs as well as plant species. The position of the collenchyma in plants is found in stems, leaves, floral parts, and fruits. It is also found in roots, especially the aerial roots, because aerial roots needs to support some mechanical type of the tissue. So, collenchyma is produced in such type of the organs or the tissues from where the aerial mechanical support is required. In stems and leaves, the collenchyma characteristically occurs in peripheral positions. It may be present immediately beneath the epidermis, or it may be separated from the epidermis by one or more layers of the parenchyma, where the organ is differentiated into ridges and furrows. Then it is present below the ridges. The structure and the shape of the collenchyma vary according to the type of the plants as well as the organs from where it is located. It may be elongated and polygonal cells with tapering ends. They are longer and narrow and evenly thickened wall cells. Extracellulose is deposited at their corners. Cell walls are composed of the cellulose, pectins, and hemicellulose, and a high percentage of water. The cells have living vacuolated chloroplasts. Cells are uninucleate and chloroplast is absent. The chloroplast is also present. It means that the collenchyma also support or perform the function of the photosynthesis. It may have or not intercellular spaces. The cell wall of the collenchyma is the most distinctive features of the collenchyma cells. The thickenings are deposited unevenly in a manner somewhat variable in different groups of plants. As seen in the cross-section, a common form of collenchyma shows the main deposits of the cell material in the corners where several cells form together. For example, in ficus, vitis, morus, beta vulgaris, and cannabis, such type of the cell walls have been observed. The walls of the collenchyma mainly consist of cellulose and pectic compounds and contain water. The collenchyma may or may not have intercellular spaces. In the absence of the intercellular spaces, the corners where several cells meet frequently show prominent accumulation of the pectic substances. These accumulations may not fill the space completely, but protrudes in the form of the warts and the choroid structures. The cell contents or the cell organelles or the chemical composition of the cell of the collenchyma. Collenchyma cells have living protoplast as maturity. They have or occurs in variable numbers. They are most numerous in collenchyma, which approaches the parenchyma in form and function. Collenchyma consisting of long narrow cells, narrowly only a few small chloroplasts or more. Tannins may be present in collenchyma cells. Collenchyma walls contain over 60% water based on fresh weight, over 200% based on the dry weight. Heating of the collenchyma destroys the ability of walls to absorb water. When collenchyma walls lose their water under the influence of the dehydrating agents, they shrink immediately or visibly. Types of collenchyma. According to the type of the wall thickening, there are three main types of the collenchyma are present. Number one, angular, lamellar, and third one is lacular collenchyma. The angular collenchyma, in this type, the thickening is primarily longitudinal in the angles or the corners of the cells. For example, the stamens of the datura. Cell lumen is almost circular in cross section when observed under the electron microscope. The lamellar collenchyma, in this type, the heavy thickening is deposited mainly of the tangential walls and in the radial walls of the cells. For example, the stems of the sambucus and the raphe. The lacunar type of the collenchyma has been observed, and in this type, the thickenings are mainly deposited around the intercellular spaces. For example, the aerial roots of the monstera. The last one is the functions of collenchyma. For example, over here, you should read the collenchyma. The collenchyma gives strength to plants where it is present. Sometimes it may have chloroplasts or the photosynthetic activities. So it performs both mainly mechanical or nutritional as well, such type of the functions. It constitutes the living pressible tissue which has a considerable tensile strength. These basically three different types of the collenchyma are present: the angular, lamellar, or the lacunar collenchyma. You can see in the diagram in the photograph the wall thickening, that is black, light purple, or the dark black observed under the microscope. However, according to the wall thickening of these type of the cells of the collenchyma, the cells of the collenchyma also differ.